This is our September meeting of New Mexicans for Science and Reason, NMSR. And we're delighted to have Kevin Folta join us. He's coming all the way from Florida. He is a professor of horticultural sciences, and also he does a lot of molecular biology at the University of Florida. And he's also a fellow of the Committee for Skeptical Inquiry. And I met him at a SciCon convention a few years ago, gave a great talk on GMO and all the myths surrounding GMO. But today he's going to talk about myths surrounding glyphosate. So I think he's going to speak for a certain time, maybe up to an hour, and then we'll have a Q&A period. So let me, I'm making Kevin a co-host. So if he needs to share some slides or movies, you'll be able to. Okay,
all right. So there, how about that? Can you hear me?
I can hear you now. Yeah.
All right. Yeah, I do this an awful lot lately with these online meetings, thanks to COVID, and then teaching mostly an online class audience, which is really a lot of fun. So let me see if I can share my screen effectively here. This is always the hard part because it puts the screen I want to project on the, I know what to do. Let me switch this. All right, that's step one. And then I make this small, and then I go to share screen, and then I share this. That's what I want to share. All right, that looks good. So can you see that okay in terms of the
looks good.
Okay, very good. So this is really a nice invitation. So thank you very much for having me on. A couple of reasons is that it really forced me to sit down and compile a lot of what I've been talking about this herbicide over the years. Herbicide that really has captured public attention, and just have a chance to put it all together in slides. This is the first time I've put together this particular slideshow, and it'll have, if there's any questions or anything as we go through, or any jumps in logic or anything that doesn't make sense, please stop me as we go along. That's just fine. So what we'll talk about today is glyphosate, which has really grabbed the headlines in the last 3 or 4, well, I should say 5 or 6 years. And what it is, where it comes from, how it's used, its potential risks and benefits. And hopefully this will give you a good sense on how you can communicate about this with others. The other thing I'd like to show you, which is of interest to everybody who deals in science and reason and skepticism, is how the science is misrepresented and how media has a role in really expanding and exaggerating what this really is and what it really isn't. So I'll start out by talking a little bit about what's important to me. I'm a professor, as was stated before. I work in molecular biology. Here, let me move this over. Oops, I lost that, didn't I? Excuse me for a second here. I got to have this up here somewhere. Hang on. This is, sorry. Where's my Zoom? Okay, that's there. I see. There's that. Okay. Sorry.
We've all been there, Kevin.
Well, I do this every single morning. I teach a class where I have to have an online session, and I really would like to have this in a place where I could kind of look at the slides as we go along. And so you might be able to see that now. It should say, "Why do I care?" Or are you seeing the presentation version?
We're seeing a big window, "Why do I care?" and then a tiny window.
Okay,
the same. Next animation. So it's got the next.
So here's what we do. We swap screens. Oops.
Hey, that looks good now.
Now that looks better. The problem is I can't. All right, we'll deal with it. We'll make it happen. So here's the story.
We can handle the other.
Yeah, we'll make it work.
Okay.
So why the first important thing in these communication exercises is why do I care? And I teach about safe food production. I'm embroiled in this at home. I'm interested in farmers being profitable. I'm concerned about the environment. I teach science because I really care about technology, sustainability in food production, and how that can reach the developing world. It's something very important to me. And I've been a professor for 20 years. very interested in the misinformation around food and farming. And I also use glyphosate. I've done research on glyphosate. I understand what it is. I know its risks and benefits. I wear appropriate PPE when I use it. And it's important to me because my wife is a farmer and we farm. And I live in a beautiful place. We raise animals. And I would never let anything happen to them or to us. And so I understand what this stuff exactly is and its relative risks. And I'll kind of tip my hat by saying I have no reservations in using it. So it only fuels the interest in talking about what it is and what it isn't. And so what is it? This is a non-selective herbicide. So when we say that, we mean it kills all plants. And I'll put an asterisk by that. We'll talk about that in a bit with environmental hazards. It kills every plant that it touches. It's a very potent herbicide. It was invented in the 1960s. It really started being used widely as an herbicide in 1974. Low toxicity to animals, but an extremely high toxicity to plants. And this stuff is really, really effective on killing plants. If you spray it in a field and there's a little bit of wind, you can affect the plants the next field over. Just a little bit of aerosol. Very, very potent herbicide. And it's the low toxicity we'll talk about in a moment. The mechanism of action is that it kills weeds by interfering with metabolism. And we talk about all herbicides here, not just glyphosate. They have to target something that's specific to plants. You ideally want to affect photosynthesis, or you're not going to affect animals, or growth, or metabolism that might be plant specific. And it's this final area of metabolism that glyphosate is an all-star. It binds into a specific part of plant metabolism that interferes with the way that plants create proteins. And with this is very effective as an herbicide. And what it does is it interrupts synthesis of amino acids. Now, most of you know that these are basic building blocks of proteins, and proteins form all of the structural and catalytic, well, most of the structural and catalytic functions of cells. So our enzymes, the proteins that make up the places where proteins are synthesized, that help replicate DNA, that control how cells divide, all of these functions are regulated by proteins. And proteins are critical for normal cellular function. So proteins are made up of amino acids. So in this figure, you have amino acids represented by different colored dots because there's different amino acids. And those different amino acids are put together by the cell and to make peptides, which fold into organization structures of proteins. And these are the shakers and movers of the cells. These are the molecules that get things done, that make the structures that do the work. And amino acids are the building blocks. Glyphosate specifically interrupts a certain set of amino acids from being synthesized in plants. And if you can't synthesize them, you can't make proteins. You can't make proteins, you can't repair the photosynthetic machinery. You can't grow. You can't create new molecules. You hinder metabolism. So glyphosate works by interrupting the synthesis of amino acids, and that's why it works. It's a very unique small molecule that exists nowhere in nature necessarily. It's a synthetic molecule that is very similar to amino acid called glycine. So glycine is a very close analog and interrupts the synthesis of amino acids. So this is the technical description. So pathways, bear with me. You basically start with some stuff and you make amino acids. So amino acids, tyrosine, phenylalanine, and tryptophan on the bottom. Tryptophan you recognize from talk about Thanksgiving, and the one that's the precursor to serotonin and other molecules. Phenylalanine you might recognize from the back of your Diet Coke can. The Diet Coke contains phenylalanine that can upset phenylketonurics who can't metabolize it. So these are amino acids that are present in just about every protein. And glyphosate blocks this enzyme, 5-endopyruvyl shikimate 3-phosphate synthase, EPSPS, we call it. It blocks EPSPS from turning this stuff into this stuff. And when you block that synthesis, the plant can no longer make the things it needs. And we know that glyphosate binds into this enzyme with extremely high affinity. It binds it and doesn't let go, blocks this pathway. Without this pathway, plants can't survive. The nice part is that humans don't have this pathway. Many bacteria, fungi don't have this pathway. So it makes it very specific and selective to plants, which is really one of the reasons why it has such low toxicity in terms of animals and other non-target organisms. So when it was introduced in the 1970s, this was thought to be a godsend, a once-in-a-century herbicide that has extremely low toxicity. You can find negative effects at extremely high doses in live animals. So if you gave them 5 grams per kilogram body weight, you could find an LD50, or the dose which is lethal to 50% of the organisms that are being studied. And the 5 grams per kilogram is really high. I mean, that's it's for a 100 kilogram guy like me, it's 500 kilograms, which is about a pound of glyphosate of the raw material to give me 50% chance of lethality. So relatively well tolerated. People have tried to commit suicide by drinking it, mostly in the developing world, and it just makes them real sick. It doesn't kill very easily. By the US EPA, it's called Category 4, practically non-toxic, non-irritating, and it is consistently reevaluated by international regulatory bodies. Re-evaluation and consistent reapproval every 5 to 10 years. The European Food Safety Organization has recently looked at it. 11,000-page study, again, plans to re-register it in the EU. Health Canada has recently re-registered. The EPA re-registered. So these things are ongoing and cyclical and continually incorporate the best new data in the most recent reevaluations. This is reflected in a consensus among regulatory bodies throughout the world, whether it's Germany, Japan, New Zealand, Canada, USA. We all have found that this compound does not cause significant toxicity at the levels used. It is no evidence of it being carcinogenic that has been detected by international regulators at levels used. Okay, so this is a really important point to make because of the controversies that swirl around it. And we'll touch on those in just a moment. So why is it important for farmers? Weeds are a problem. You know, it's impossible to farm without dealing with weeds. And as someone married to a farmer, I see what we do here to get rid of weeds, and it's mechanical removal. It's digging them out by hand or with a plow. And weeds are yields and cause a loss of the effectiveness of fertilizer and water, who are stolen by weeds rather than the crop plant itself. So weed control in farmer fields, and especially in large agronomic crops like corn, soybeans, sugar beets, it's always been done by tilling and selective herbicides, meaning herbicides that won't kill grasses. So things like corn, wheat, these are grasses that you can spray with certain herbicides that only will kill broadleaf weeds and leave the grasses standing. And this has always been how they were killed before. So when glyphosate came along, it was very helpful because if there was a way that you could use glyphosate to kill weeds and leave the crop standing, then possibly this would be an excellent innovation that would help farmers. And that's where genetic engineering really took off in the 1990s, because scientists were able to engineer in a form of the EPSPS enzyme, that enzyme that binds glyphosate, that didn't bind glyphosate. It was a naturally occurring variant that glyphosate didn't bind and didn't care about. So if we went to that that pathway, let me go back to that pathway. If we go to that pathway, if this enzyme is replaced by a different version, a naturally occurring version that doesn't bind glyphosate, now this pathway would proceed. So it was replacing the plant's own native one with one that wasn't affected by glyphosate. You could give glyphosate all day and it didn't affect this pathway. You could engineer that enzyme into plants genetically through those techniques. And now I have plants that you could spray with the herbicide and they would survive. It was a really important innovation. And the reason it worked was because using a GE and by introducing this enzyme, you allowed farmers to grow plants, grow their crop, drive over that crop and spray with glyphosate, kill the weeds, but leave the crop. And it was always applied very early in crop development. So you would have corn or soybeans, whatever, that were just emerging and just starting to grow, along with all the weeds. Farmer drives over the top, sprays with glyphosate, and only leaves the crop. And then at that point, the crop could outcompete the weeds by growing very rapidly, shading them out. So this was an excellent strategy for farmers that had a lot of very good benefits. And it worked, kind of going back to where I was before, because there was a special enzyme that was in what they called Roundup ready crops that you could spray with the Roundup, which is the brand name of glyphosate. And this would survive, whereas the rest of the plants would die. And it gave this plant the ability to survive this treatment. Now, as I mentioned before, this is an extremely potent herbicide that you would be spraying essentially 750 grams of active ingredient over an acre. And as I mentioned before, the LD50 was 5,000 grams, 5 grams per kilogram of body. Go ahead again, 750 grams per acre. The LD50 is 5 grams per kilogram of body weight. So you could see how it would take essentially the amount that it would take to cause 50% lethality in acute treatment was about what you were spraying over 2/3 of an acre. So very, very potent to plants, very, very not potent in acute toxicity to animals. And again, the mechanism of resistance, you replace this enzyme with a version that doesn't bind glyphosate and allows this pathway to work in plants that are sprayed. It's kind of like taking the old incandescent light bulb out of a light fixture and replacing it with an LED light. It does exactly the same job, only is significantly different in terms of its composition and technology. It's taking out this enzyme and replacing it with one that does the same job, but doesn't bind the herbicide. So the pathway continues. It was an amazing innovation, and farmers loved it for a lot of reasons. It was introduced in the corn, canola, soybean, sugar beet, alfalfa, and cotton. And what you may find surprising, is that there's really just 4 that contribute to the human diet: corn, canola, soybean, and sugar beets. Corn gives us high fructose corn syrup, corn solids like starches, corn oil. Canola gives us canola oil. Soybeans give us soybean oil, soybean solids. Sugar beets give us sugar. And these are the 4 places where this has been introduced. There is no herbicide or glyphosate resistant grapes, apples, tomatoes, all the other things that people claim are loaded with glyphosate. It just isn't there. And we'll talk about where glyphosate enters the diet in just a moment. But these are the 4 where it's been engineered in. This is for cattle feed. This is for fiber, but also maybe some cottonseed oil gets in the food. But in general, it's just these 4 items. And it's just the ingredients from these 4 items. What's really important to note there is if you make sugar from a sugar beet that's been treated with glyphosate early in its development, there's sugar and no glyphosate. There's no DNA. There's just sugar. Soybean oil contains soybean oil. There's no glyphosate there. That's soybean oil. There's no DNA. There's nothing else. So these are the ingredients that come from these crop plants. And that's a really important point to make. So why do farmers like this? If you use glyphosate on your crop, as I mentioned before, you spray it early, the weeds go away, the crop remains. And the reason it's important is because you use less fuel, less labor. You're not doing a lot of tilling. You're having a safer product for applicators than atrazine or some of the other herbicides that used to be used. Lower cost. Glyphosate used to be extremely cheap. That's changed a lot. And if you have less weed competition, you have better yields. That's kind of marginal. That's not a real big benefit. The big benefit is being able to control weeds at a low cost. The big advantage for the environment is that it enables low till or no till farming or conservation tilling. And what that means is you're not disturbing the topsoil. And when you don't disturb the topsoil, it's retained, it stays in place and keeps the CO2 that's trapped in this topsoil in place. It doesn't physically disrupt soil and the organisms in it. So there's a lot of benefits that way. When you talk to farmers in places like Australia, where they have tremendous winds that would kick up over farmland in Victoria, they would talk of these gigantic dust clouds that would move into Melbourne in the 1980s from the farmland that was just west. And when glyphosate started, that stopped because you weren't disturbing the soil and weren't blowing off the most rich topsoil that was there. So this is a real benefit for farmers and the environment. For every benefit, though, there are a number of risks and a number of things we have to be careful about. And most of these effects are ecological. We know, no doubt, that when you use an effective strategy to control weeds, weeds will evolve around it. You spray it every year, nature finds a way. It's like antibiotic resistance in medicine. When you take antibiotics regularly without being careful, you develop antibiotic resistant bacteria. The same thing happens with weeds. And since glyphosate's introduction, there's probably at least 35, maybe eventually 50 types of weeds that are now resistant to the herbicide. They make the farming less effective and they cause other problems with respect towards cultivation of herbicide resistant crops. So this is a big deal. Glyphosate can have negative effects on aquatic, or I shouldn't say negative effects. They have effects on aquatic communities. You see changes in aquatic communities, what is present in terms of the organisms, the insects that are there. That's been outlined very well and underlies why we are very careful about using glyphosate around aquatic environments. It's been very well documented that there are developmental changes in fish and fish that show signs of toxicity when in glyphosate. So you bathe them in glyphosate, essentially they're in the water with the herbicide, you can see some effects. And I believe those are real and they're minor, but they happen at higher concentrations generally, but they are there and they are a caution that must be heeded when using this compound. We know that one of the big problems in conventional farming is that there's been depletion of diversity around fence lines and pastures, that when you're using glyphosate to essentially clear the soil of the plants that are there to farm and clear fence lines like we do, we have electric fences that protect our livestock. And we use glyphosate to break the connection between the electric fence and the ground. And it removes all of the flowers and things that grow there and give less forage for bees and butterflies, the other animals that are here, which means we have to set up a separate space to grow those forage natural plants. So we do that. I don't know that everybody does. And so it is a potential downside of a very convenient herbicide. The other one is that in places like the Amazon, soybeans are extremely profitable. They're genetically engineered to resist glyphosate. And so farmers are tempted to expand their acreage, removing trees, moving in the sensitive areas, moving into areas where maybe species exist that are threatened, who knows? But taking that area and turning it into cultivated area so that we can feed more people, you're now sacrificing area that was previously a natural area for biodiversity. And so this is another risk of glyphosate. You create a convenient tool to help farmers farm profitably, and they use it. And it works so well that they expand their acreage, and that can also threaten various aspects of our environment. So the effects are negative, are ecological, well documented, well understood, and precautions are taken accordingly. So that's a really important point to make. In terms of formulations, and we'll get to negative health effects in a second, formulations, this is applied as the active ingredient, glyphosate, plus a surfactant. And surfactants you can think of as soaps. They are the detergents that break surface tension. So you put that drop of dishwashing liquid into a sink full of oil, you see it scatter because the surfactant captures oil. It breaks surface tension. And when you're putting an herbicide on plants, you want to use a surfactant because it breaks the waxy cuticle and allows an herbicide to penetrate. So glyphosate plus its surfactant, which is POEA, which is polyethyl oxalated toluamine, is under the brand name Roundup. Now you can find glyphosate not as Roundup, but as a whole bunch of other preparations. The smart farmer buys it in generic form. I pay about $50 a gallon for the stuff as a 53 or 41% concentrate versus buying a 1% consumer grade preparation for around $30 or $40 nowadays. So it is present in a generic form with different surfactants. They all work great. They all kill weeds. So when we talk about Roundup, we're talking about the name brand. I do not endorse one over the other. I don't use Roundup. I never would. It's too expensive. I use the cheap stuff and it works great. So what's important is that glyphosate is present in 750 different brands of weed control formulations. So that's really important to make this distinction. When we start talking about the legal actions that have been taken, they're suing Roundup makers, not the makers of glyphosate. It's a really important point to make, and we'll get to that. We know that this stuff is persistent in the environment with a half-life in soil that varies from 1 to 280 days. That's a range. It breaks down to something called AMPA, which is present maybe even longer. And we know this is degraded by soil organisms, but that's totally dependent upon the environment itself. It breaks down with water, higher temperatures, with light, different soils, different soil organisms. It really varies. So when we're talking about glyphosate and its persistence in soil, it's all dependent upon the environment that's there. So the question always comes up, well, if I spray plants in the soil and I get some on the soil, how does that affect next year's crop or the next crop I plant? Very, very limited effects for two reasons. One is because glyphosate is a foliar herbicide. It works when you get it on the leaves. The surfactants allow it to penetrate the leaves. If you get it on the soil, it's essentially wasted. It goes on the soil. It's not taken up well by the roots. It's also diluted. And if you don't have the herbicide along with its surfactant, it is less effective. We've done experiments in my lab where we grow them in petri dishes, grow plants in petri dishes containing glyphosate, and it takes probably 10 times the glyphosate to affect a plant through soil or media uptake in our case. So very, very important to get it on the leaves. Farmers who spray it on the soil look at it as wasted product. So it's very important to keep it on leaves. Pharmacologically, moves through the human body very quickly. It moves through the digestive tract. 95% is lost in urine and stools within a day or so. Well, no, a couple days, 95%. 5% is metabolized by a specific enzyme. We know that it's easy to detect. And when we detect glyphosate in human exposure, we look at human urine. It moves through the body. And so we know that it's in the urine. We can guess how much you've been exposed to by how much is present in the urine. And the methods for detection in urine are very good, very sensitive. So we can get a very good handle on how much is moving through you by looking at that. And what's important to note is that this is present in the urine and in the stools, which is where you want to find an herbicide. You don't want it accumulating in the body. DDT was an insecticide that was notorious for accumulating in biological organisms and causing effects because it accumulates over time. Glyphosate does not accumulate in the body. It's not fat soluble. It's water soluble. It moves in, moves out. So any exposure that you have as an applicator, as a farmer, as someone who may consume it in one way or another, it moves through you. And this is a very important point to make when we start looking at some of the claims. We know that it's detected in urine. And the nice part is, the really good part is, we can detect almost none. So something that's present on the edge of nothing, we can tell you it's there. And so let's talk about that. 44% of urine, these are published data. I should have mentioned, I should have cited the first one. 44% of Europeans who are surveyed show, 44% show that it's detectable at a level lower than 1 part per billion. And a recent assessment by the CDC in the US says 80% of the US population has detections of 2 to 3 parts per billion. That sounds kind of alarming, 80% of people having detection. But what is a part per billion? That's equivalent to 1 second in 31 years. So what it says is, to me as a scientist, is wow, or something that's almost not there. And it also says there's no risk associated with levels that low, or very minimal risk, exceedingly minimal risk. It also says it really is a testament to how safe the food is. That if we can monitor and detect these levels that are so low, it really says we're doing something right, that we're monitoring well, detecting it well, and showing that it's almost not there. For most people in the population, they want 0. And it's impossible, almost impossible for us to say there ever is 0 because we're so good at finding it. It's pretty remarkable. So what's the presence on food? And I mentioned this was used on a very thin layers of GE crops. It is usually processed long before, so it's applied before food is present. So the soybeans, the corn, the sugar beets, stuff that goes in your diet, it's sprayed before that stuff is on the crop. You don't spray the soybean pods. You don't spray the corn cob. That just isn't done. It's hard to find reliable measurements about the claims of what's there. The ones you can trust are from USDA, from CDC. I think you can trust those numbers. But a lot of stuff online that says it's present at higher levels, that they're using inappropriate tests. So it makes it very hard for us to separate what's real and what's not. As I mentioned before, these are the ingredients like high fructose corn syrup, starch solids. And one of the things that we have to keep mindful of, even though wheat is not genetically engineered, there's no GMO wheat, glyphosate is applied as a harvest aid. And what that means is that in some circumstances, under certain climatic conditions, when we've had rain or whatever we've had, it's important that farmers dry the wheat evenly. And they will use glyphosate to burn down or to dry as a drying agent to ensure that all the wheat is dry at the same time. It helps harvest a more uniform crop that is more marketable. So this is a rather recent application that results in detection in glyphosate on non-GMO crops like wheat. And that's another thing that there's been a lot of criticism of. I don't know how much gravity we should put into that, but keep that in mind. So controversy. So we know for a long time that people have been opposed to genetically engineered crops. And in the last 10 years, glyphosate has really become the proxy for GE crops. If you couldn't scare people away from genetically engineered crops, maybe we could target the herbicide because it's got the suffix "icide," right? It means to kill. And you could scare people with chemistry more than genetics. And I think that was the tactic that was used by folks who were opposed to these technologies to really use inappropriate science to criticize what this technology is and isn't. This really was fueled by the IARC decision in 2015. The International Agency for Research on Cancer, which is an arm of the World Health Organization, decided to list glyphosate as a class 2A probable carcinogen. That was an extremely controversial assessment. And when you look at how they arrived at that decision, you really scratch your head as a scientist, because the data and the papers they used to make that decision really didn't come to that conclusion. There was one paper that had some data that suggested it was carcinogenic. Others, which were just trending that way, didn't statistically back those conclusions. I could talk about that all day. But the most important part is that that was a hazard-based decision, not a risk-based decision. What's the difference? There are, the great white shark is a hazard. I know it's potentially dangerous. It's something that could kill me, but sitting in my home in the middle of Gainesville, Florida, I have very low risk of being killed by a great white shark. So the IARC decision was a hazard-based decision. Could this potentially be a carcinogen if you were exposed in high enough ways, high enough levels in certain ways? And they said, maybe. They didn't say definitively, they said possibly. They didn't say that it was a risk in the levels in which you were exposed as an applicator or as a consumer. There were conflicts of interest on the IARC panel. There were people who are already recruited by law firms that would later play a central role in the lawsuits against the companies that manufactured Roundup. And other WHO, World Health Organization agencies like the Joint Pesticide Task Force did not agree with these findings, as did the WHO. They were explicit in saying there is no evidence based upon the levels used that glyphosate is a carcinogen. And that was like, it's very explicit by the WHO, who looked at the IARC decision as being potentially politicized, really overstepping the data, and something that really kind of tarnished the reputation of IARC and the World Health Organization. The decision that they had, that they did come down with, said limited evidence in humans for the carcinogenicity of glyphosate. They said there's strong evidence that exposure to glyphosate in certain glyphosate-based formulations is genotoxic. Therefore, we'll call it probably carcinogenic, along with red meat, hot beverages, working as a barber. These are other things that they suggest are potentially carcinogenic. So IARC, goes in their group 2A. That's a really important distinction to make. Probably carcinogenic. We don't know for sure. Don't have any evidence that it is, but we'll say probably is, based upon some data that suggest there is a possibility of causing the hallmarks of cancer. Very, very flimsy data that supported that. If you look at what they mean by that, group 1 is tobacco, the sun, alcohol, bacon, processed meats. These things they know are carcinogenic. We know there's higher incidence of cancer that occur epidemiologically. We know the mechanisms by which these things cause cancer. Probably carcinogenic, frying certain foods like potatoes, steroids, exposures to working as a hairdresser, red meat, and glyphosate. Maybe, if you have certain exposures, could be carcinogenic. So this is a really important distinction to make, because online you'll find this stuff causes cancer. Court cases will say causes cancer. There's no evidence to support that conclusion. In fact, the agricultural health study, which is a study of over 54,000 applicators since 1993, a study that has followed them together, says there's no association between glyphosate and tumors or lymphoid malignancies. And so the best studies say this does not exist. So where do those data come from? When you look at all the studies, so each one of these dots represents a different study on glyphosate and the incidence of cancer. And many of these are in farmers and applicators, where they'll give them a survey that says, tell us about the diseases you suffer from and tell us about the chemistry you've used. They may have used dozens of other chemistries. They may drink a lot. They may smoke a lot. They may all the other things that they could do. But all we can say is that when you look at the associations, of no association, which would be right on the line, and reduction of cancer or increase of cancer. And this is a one-fold, 10-fold increase of cancer. This is probably about 3% increase. All of these are pretty minor effects on cancer, whether they increase or decrease. And here's non-Hodgkin's lymphoma, the one that people claim affects cancer the most. And you can see a little bit of a trend there. But a lot of these studies that have been done here have been done with higher resolution and do not support that association, nor does Andreotti, the highest resolution study that we have. So the same argument can be said, well, look at the reduction in kidney cancer and lung cancer. Glyphosate cures or limits kidney cancer. We would never say that. But this is where these hunches come from these early reports that suggested this weak association that then could be built upon by the folks who wanted to build upon it. Health Canada says no pesticide regulatory authority in the world currently considers glyphosate to be a cancer risk to humans at the levels at which humans are currently exposed. And this came from a special agency that was convened after the IARC panel that assessed glyphosate one more time and did this with independent scientists who had no conflicts of interest because they wanted to get to the bottom of it, and this was their conclusion. Where do we see negative effects and where were these claims made? And these were made basically on in vitro studies, that when you put glyphosate in vitro at high levels, you could see effects on cells which were reminiscent of DNA damage that would be indicative potentially of a possibility of increasing cancers. We knew that in vivo, when you fed animals, negative effects like death could be seen at high dosages, other effects, and we knew about effects in aquatic animals, aquatic microorganisms and insect communities. But those were the negative effects we knew about. But that didn't stop certain elements from showing us data that suggested that glyphosate was dangerous. And 10 years ago, this week, this paper came out where they claimed that GMO crops plus Roundup, GMO feed or Roundup alone caused tumors. And this paper by Seralini et al. Was highly controversial upon release. These animals are in a state that is ethically unacceptable to animal researchers. The one thing that you'll also notice is that something is missing from this figure. What about the rat that received no GMO corn or Roundup? Well, guess what? It got tumors too. These are a kind of rat called a Sprague-Dawley rat, which by 2 years has massive tumors. They left that one out conveniently, which probably would have taken a lot of the bite out of this figure. It somehow passed peer review, somehow was accepted by the journal, and then later retracted. Very controversial, and lots more discussion on that if you're interested after we get done here with the talk. There's been a lot of assessment of in vivo data, lots of assessments that were done. We see effects at high levels, but never at low levels, especially at exposure levels for occupational or dietary levels. It just doesn't happen. There's lots here. I'll leave this. I won't go through it because there's lots here, but you can look at the levels at which they were using, very, very high levels. And this should, if anything, confirm the safety of this compound, that when you give it huge amounts, you can see effects, but you're not seeing them at lower dosages. The bad news is that glyphosate was used as that proxy for a war against seed producers. And this is a political thing, not a scientific thing. And that was really important for us to note. We see papers online that were grabbing headlines. This paper drives me crazy. It was published in a journal called Entropy. There are no original data in this paper. It's a review of cherry-picked pieces of evidence from other papers done by an MIT computer scientist who is not on faculty, who uses the credibility of MIT to bolster her claims. It gets everywhere in social media. MIT says, no, MIT steps away from this. And even the editor of the journal, they say, this showed biased citation and probably problematic. No evidence of fraud, misconduct, all that stuff. That's probably true too, because all it did was cherry pick data. Very, very impactful. The same author appears on Fox News talking about the danger of the COVID vaccine. She makes a living publishing books and writing articles that are scientifically inconsistent with the scientific consensus, yet grab headlines and make an awful lot of noise and make my life more challenging. You can see she publishes everywhere, and she's one of the main people who is pushing these claims. She gets published in very low quality journals, predatory journals, journals that maybe have lax review, and none of this contains original data. It's all data that have been inferred from other reviews. It's really problematic for scientists to push through. So can you trust the claims about detection? In some cases you can. There's a detection kit that's available. You'll see this online. If you see that they're detecting it in food or water or wine or beer or whatever, you can sometimes trust those data. Detection kits are there, but they work only with urine and water reliably. Not with breast milk, not with water, not with wine, not with beer, not with crackers, not with anything else. Those we have to go to LCMS. And with LCMS, we can be very confident in what we're seeing, but it's an extremely laborious, expensive, time-consuming process. But we can get it. And that's where the good numbers come from. So if you're ever concerned about a number you see, look at what they're claiming to detect, and then look whether it's using this whether it's using this ELISA-based method, or whether it's using LCMS. And if it's using LCMS, I think you can generally believe it. And we'll kind of hammer
Kevin?
through. Yeah.
What does LCMS stand for?
Liquid chromatography, mass spectroscopy. You can look at the molecular weight and characteristics of a molecule that's separated using liquid chromatography. These two things coupled together are extremely potent and are very sensitive to be able to detect the glyphosate. Yeah, thank you for a good question there. So when you looked online, there's been a lot of discussion of it's in breast milk, and you've seen a lot of claims made to that, a lot of sensationalism, scare journalism. But they use this ELISA-based kit, this kit that you can't use it on breast milk without creating false positives. You can do it with LCMS, and it was done, and they didn't detect it. And so this got headlines. It was done by an activist group in California. This was done by a professor who's an expert in breast milk. Didn't find it, didn't get a headline. This did, that didn't. So you can see where the problem is amplified by even the traditional media. Beer and wine, they claim to detect it using this kit at 51 parts per billion. Again, on the edge of nothing, 51 seconds in 31 years. Probably just a false signal of the kit. Organic wine, which you can't use glyphosate in organic cultivation. They're still claiming to find 5.2 parts per billion, maybe less than the conventional. But seeing that tells me you're detecting this in a negative control where you can't use glyphosate. You're still seeing it. It says there's something wrong with your assay. These have been all over the web about different beer and different products that contain it. Still exceedingly low levels on the edge of detection. Tell me maybe this is probably not reliable data. And the funny part is, as I mentioned, I show beer and wine here for a reason. I mentioned before that alcohol, ethanol, is a class 1 carcinogen. And so people are making claims about its presence in beer and wine and how this is a problem. But I made this a while ago. You're saying that there's 1 part per billion or maybe 5 parts per billion of a probable carcinogen contaminating your 130 million parts per billion of a known carcinogen. And it's just kind of the irony of this. This is considered to be risk, whereas the real risk comes from the wine itself, not from this chance contaminant that may or may not be there. So let's start heading towards the end here. I want to show you another couple of examples of how this gets blown up in the media. This paper came out recently at the end of July. Glyphosate infiltrates the brain and increases pro-inflammatory cytokine TNF-alpha, implications for neurodegenerative disorders. Excellent paper, good data. I disagree with implications for neurodegenerative disorders. They fed massive amounts of glyphosate to mice, and they looked to see if they could detect it in the brain, and they could. they could detect tiny amounts in the brain when you fed mice massive amounts. But this is shown in the media here, Arizona PBS, pesticide research links chemical compound to brain disorders. The research did not do that. It said, if we feed mice massive amounts, can we see it in the brain? Yes, you can. They didn't show that the mice developed disease, didn't show mice had problems with memory, which you could do those tests. It didn't show mice had any problems neurologically. It just said that it was in the brain. But it took off. Glyphosate weed killer crosses the blood-brain barrier linked to Alzheimer's disease and neurodegenerative disorders. That is not true. It is not what the study showed. But when the authors put implications for neurodegenerative disorders and then meet with reporters and say, well, maybe this is it, the authors probably had their best interests at heart and were just being honest by saying, maybe this is a hypothesis to test. But when you tell that to a reporter who's hungry for a story, it gets blown up. And then the internet goes nuts from there. Now, all of a sudden, this is automatically linked for the rest of humanity, the rest of the time, to neurological disorders, even though that was not shown at all by the data. And this is why all of us in a skeptical community, in a scientific community, have to be active in social media and say, it's not exactly what that claimed. And when you're encountering these kinds of claims, saying simply, they had to feed the mice massive amounts, it didn't show a connection to neurological disease. Because the majority of people who are out there are confused about what they're reading about these chemistries and about GE crops, and they don't know who to trust. And they trust these sources. And scientists aren't stepping into those conversations. I do it all the time. I monitor the hashtag glyphosate, and I just keep it real. And I direct people to real information, good data that hopefully can at least participate in the controversy. Here, natural news study can cause Alzheimer's disease. No, it doesn't. I mean, look at how far that's extrapolated from the real claim. A possible link to Alzheimer's in the Arizona Journal Star. The actual study, a mouse experiment where they gave 500 milligrams per kilogram for 14 days. 500 milligrams per kilogram for me as a 220-pound guy is like taking 200 aspirin-sized tablets per day for 2 weeks, and then saying, can we detect it in your brain? Damn right, you better be able to. I mean, I'm going to be peeing this stuff out like crazy, but you probably could detect it in a lot of my organs because I've been saturated by being forced to eat 200 aspirin-sized tablets of the stuff per day for 14 days. It does not show that it's causing Alzheimer's. It does not show neurological disease. It says that when you pack a mouse full of the stuff, you find it in the brain. That's all it says. But you can see how the media and social media and activist media create a new story from what actually was a good result. We saw this happen in many places, and concerning how little we understand its impact on the nervous system. It's amazing what we're seeing. And I'll wrap up by talking a little bit about the legality. Now, how glyphosate infiltrates your brain, you can now find a lawyer if you have Alzheimer's disease. Dial 833-LETS-SUE because you may have a legal claim. This was found on the web today. Other studies, disturbing weed killer in 80% of human urine. I talked about this before. This is in The Guardian. What they didn't tell you in this article was that it was detected at 200 parts per trillion, or about 3 minutes in the last 31,000 years. So it can't give you any sense of risk there. It's thousands to millions of times below the level at which it's important, or which it could have physiological effect. Yet here they're talking about it's present in your urine, better be nervous. Another study showed that when it says Roundup linked to convulsions in animals, not really. This went everywhere a few weeks ago. When you electrocute a worm, it has a convulsion. If it's electrocuted in the presence soaked in Roundup, that convulsion lasts a little longer. Good study, perfectly good data. But again, I trust the researchers, but the media takes it farther away. And the university media is the worst. They're the ones who said, well, now we know it's linked to neurological problems. It didn't show that. It showed that when you electrocute a worm, it convulses. The convulsion lasts longer in the presence of the herbicide. So, I don't want to talk about the lawsuits very much. I'm not an expert in this area, but it's important to note that they're not suing the manufacturers of glyphosate, the hundreds of them. They're suing Bayer, who is the manufacturer of Roundup now. Monsanto was before. They're suing the deep pockets. And a lot of this is folks who are working with this are folks who have been rallying against genetic crops and how they're used and claiming they're dangerous. So again, the herbicide being used as a proxy for the herbicide itself. So I'm going to wrap it up there. Glyphosate is a broad spectrum herbicide. We know how it works. It breaks the plant's ability to make proteins. It's been valuable for farmers. It's very rapidly adopted by farmers. I know farmers who are ready to quit farming that stayed in farming because they said, I got this tool that allowed me to continue to make a profit or break even while farming. We know that there are environmental risks. They're well described. We know how to minimize them. There's no evidence of risks to human health at levels used. Zero evidence it causes cancer. Campaign that's well targeted, well coordinated between activist communities and legal authorities. Some say even other organizations. The Church of Scientology has shown up in here that look to cash in on these claims. And it's starting to be looked at a little more critically. The last couple claims have not been favorable for the plaintiffs. So, there's no evidence of a causal association, yet these claims continue to be filed. And Bayer has settled for a certain claim to just make it go away because they'll be fighting this for the next 100 years if they don't. If you want to check out what I do on this, there's my email address. I'm always happy to answer questions. I'll answer questions today, as long as you like. I do 2 podcasts a week, one talking biotech, where we take on many issues in animal and plant biology, medicine, with respect to biotechnology, new techniques and cool new things that are being done there. And I also do one called Science Facts and Fallacies, which explores 3 new science stories every week. And we do it in kind of a hopefully entertaining way. So that's me, and I'm very happy to answer any questions. It looks like Rocky Stone. Is that right? Yeah, you have a question? Go ahead. Yeah, you just have to unmute yourself there.
Okay,
Can't hear you, Russ.
unmuted.
Yeah.
Okay, what kind of PPE do you use when you use Roundup or glyphosate? And second, my second question: do you testify as an expert witness in some of these lawsuits?
Okay, so let me answer the second one first. I've never testified with anything to do with glyphosate. I've been kind of excluded from that pool for a couple of reasons. But I have served as a subject matter expert in older questions about genetic fidelity. So I have had roles like that, and they're really a lot of fun. Never had the opportunity to testify on glyphosate, and I would welcome that opportunity. The first question is PPE that I use. I use PPE. First, I'll tell you, I use a very large droplet. So when I spray, I use a very large droplet, mostly from a backpack sprayer. I don't use any kind of mobile spraying device. We're small enough to do that. I use rubber gloves, long sleeves, long pants, boots, and eye protection, usually wear a hat. And after application, I clean up real nice and take a shower. It is, I know exactly where that spray is going. I'm very careful with wind conditions and weather, rain, that kind of thing. That's the biggest deal. Big droplet size, wind conditions. I am very confident that no drops end up on me. I put it on the weeds where it belongs. When you look at my spray patterns, you look at incidental damage on crops, you just don't see it. It goes right where I want it. Even the grass around those areas not affected. So always, it's the practices you use and the PPE are very important. Any other questions or thoughts?
Yeah, I got a question about milkweed.
Yeah.
Monarch butterflies need milkweed.
Yes.
And it seems like the herbicides can wipe out that milkweed. So is there some sort of workaround so that it would be a win-win.
Yeah, milkweed is an unfortunate loss of this technology because milkweed very easily killed by glyphosate. And so that's when I'm talking about fence lines, expanding pastures, expanding farm area, because farmers prices of commodities are too low. And to make money, you need to expand your acreage, and genetically engineered crops allow you to do that. So you expand your acreage into adjacent pastures, you destroy this natural habitat, and that is a very real risk of these technologies. So the alternative, on our space, we plant lots of wildflowers. We have lots of native stuff that grows here for native pollinators and for butterflies. It is a unfortunate collateral effect of this. The good news is that there's plenty of natural forages that are still out there. And with monarch populations in decline, some reports say it's something that we always need to be very sensitive to. So something that certainly I'm concerned about, and something that I keep an eyeball on.
I guess I didn't quite understand the answer. Is there something that can be done to really let the milkweed live in big enough patches for the monarchs to be able to migrate?
Yeah, there has to be specific forages that are established. So you have to designate specific space for milkweed forage. The other example that fell on deaf ears was creating glyphosate-resistant milkweed. But part of the word milkweed is weed. It's not a crop that you want growing in your agricultural space as a commodity. So designating specific forage spaces where butterflies could grow and do their thing, that would be perfectly fine. So that is something that many farmers have done, that many organizations have done, and hopefully will have a positive effect on monarchs.
Okay, so it is being done.
Yeah, at least in the United States. I can't say much about Mexico, where they go and habitate down there. But certainly, development and expansion of farming is a bigger risk to milkweed than genetically engineered crops themselves, which are an indirect contributor to that expanded acreage. Any other questions, thoughts? I'm a professor. I'll ask you questions if you don't ask me.
Looks like Randy has a question for you.
Yeah, Randy, go ahead. Maybe I'm not seeing everybody on the screen.
Yeah, probably not. Your comment about being able to promote no-till techniques was very interesting. I think that's been identified as a really key change that farming could do to reduce carbonization.
Yeah, and that's been a huge benefit of glyphosate, is the no-till farming, not just in carbon reduction, but also in the loss of topsoil. The runoff of topsoil, especially in the Mississippi Valley and other major river valleys, is a significant concern, because here you have a water source that's being used for irrigation and which during rainstorms you have soil loss, and being able to retain that soil is a huge advantage. And then the savings in diesel, labor, and then the carbon that's produced to do the plowing, that's massive too.
It would seem to be advantageous to promote this as a climate-friendly practice or product, and maybe change that public relations picture a little bit.
Yeah, I think so. I mean, I agree with you 100%. I think that the problem is that climate is a tricky one to motivate folks around when you're showing pictures of lumpy rats. One of them is the claims that are immediately affected, that are claimed to affect human health and family, that kind of thing, versus long-term effects on climate. It's always hard to balance that. I certainly agree that any incremental changes you can make to contribute to stem climate change are important. And the effects of decarbonization through no-till are really significant.
Yeah. Good. Thank you.
Yeah, thank you.
So I had a question about it seems like most of the opposition to glyphosate is coming not from farmers, but from basically the anti-GMO movement that has found a proxy to get people bothered about. So are there any farmers that are out there genuinely concerned about Roundup or glyphosate?
I haven't met any conventional farmers that are really against it or concerned about it. You find a lot of farmers who grow organic who want to ensure that their crop does not test positive for genetically engineered DNA, that they would say we don't want Roundup ready crops growing adjacent to organic crops because it cuts their value potentially. That's true. Some farmers worry about things like drift. So if you're growing something like tomatoes, or papayas can be very sensitive to glyphosate, you have to be very cognizant of drift that would come and potentially damage your crop. And the signatures of glyphosate damage are very, very clear. And so
what kind of drift is that? Just like wind drifting?
Yeah,
or?
physical drift,
Okay.
physical drift. So that's why when I talk about when we use it, droplet size, applying early in the morning when there's no wind and when conditions are exactly right is so important. There is a lot of there can be issues with physical drift of the aerosols. It does not volatilize, so it doesn't turn into a toxic gas cloud and move far away like dicamba and other herbicides do. It is very much where you put it, it stays. It's another nice advantage of this particular technology.
What size of minimum drops for the drop size you want, not the aerosolized?
Yeah, I can't tell you that accurately because I don't use a tractor-based sprayer. That's where you have nozzle size controls that are extremely precise and they can give you exact diameters. That's not something I use a backpack sprayer typically on my space. We grow very diverse stuff and I can walk a fence line in an hour, fill up a couple times. But droplet size being large enough where I can physically see independent droplets and watch them coat leaves.
Okay.
So yeah, it's big droplets.
All right, thanks.
If as I get to the end of the sprayer, you start seeing kind of that mist, that I stop there because I don't want that moving on crops because crops make money and I don't want them damaged by herbicide. Any other thoughts or questions?
I have some old Roundup.
Yeah.
Do you think it would be safe if I used it in a prescribed manner?
I believe it still should be very effective. This stuff is extremely stable and it's used in formulations that stabilize the active ingredient. So even if it's a little bit old, it probably still will work to some degree. It may take a little longer to kill the weeds, but give it a try on a little sample patch and see how it does. And maybe the 1X dilution. So if you buy Roundup, if you already bought it at its regular dilution, give it a shot and then maybe do an extra application later that same day. Let it dry and then try it again. And then next to it, do just one application and see if you can do the experiment that does it still work at its prescribed rate.
Thank you.
Oh, you're welcome.
All right. Okay, well, that was an excellent talk. I'm really glad Kevin stayed up late to visit us here in New Mexico.
The night's just beginning. I'm still rolling. I appreciate the invitation because I've never done a presentation on glyphosate by itself before. And it really was an impetus for me to make an overdue presentation and find the holes that I need to fill and organize this more effectively. So I really appreciate you being my guinea pigs.
We're happy to help and we're the beneficiaries of all that. So yeah, we'll post this on the YouTube and it'll be written up in our newsletter next month. And then so yeah, I'm gonna start tracking on Twitter pound glyphosate and when somebody says something really silly, I'll be able to pop this the address for this YouTube coming up and say, hey, you need to learn that you'd have to ingest a whole lot of that stuff before you get cancer.
Very good, Dave. I really appreciate that sentiment, because most people who are out there are just keep seeing the negative news and seeing the fabrications and the extrapolations, and they're not being exposed to the science. And even though we never may convince the people who are against it to change their minds, we can have a profound effect on the folks who are in the middle. And it's very important for us to be present in that conversation.
That's something I've said for years. If the scientists don't get out there and provide counter examples to all these false claims, then the general public is going to be led to think, well, there must be something to them. Nobody's saying they're wrong. So yeah, it's important to stand up and try to inject some truth into the conversation.
I had one other question.
Oh, sure.
About the litigation, and you mentioned that it's almost universally, or is definitely universally, about the Roundup manufacturer itself, Bayer, and not about the glyphosate manufacturer. Is there, and the surfactant is essentially the only other, maybe a stabilizer, so in Roundup that's not in the basic product. In any of these lawsuits, do they touch base on all of the components of Roundup versus the basic product?
That's an excellent question. Do they look at the final product versus the active ingredient?
Right.
You know, I won't jump out on a limb to answer that. I've kind of I don't have the bandwidth in my life to look through all the court documents and really look into that.
But somebody surely is doing that, right?
Yes, certainly there are people who do. And I think that there has been so much frustration in the scientific community around this and in the agricultural community, because here's something that we know has never shown carcinogenicity, but is being claimed to be a carcinogen. And then you have the company, and you can agree with this or not agree with it, that says we're going to settle this, and we're not going to claim that we have any fault, and we're never going to say it's carcinogenic, but we'll go ahead and settle with these people. So they're basically writing checks for billions of dollars. Lawyers know that this will work. They did it with talc. They did it with silicone breast implants. They did it with other stuff. And they're going to probably do it again with glyphosate with neurological disease. So there's and they're doing it now with Parkinson's and paraquat and other herbicides, because they know that this is a piñata, that if you can take a big company with deep pockets and guarantee them a decade of very expensive legal defense, it's much cheaper for a company to say, we'll just pay you off, pay those lawyers, and then make it go away, not admit to any fault. Now everybody goes forward. And unfortunately, it's a flaw of the legal system. Jury of 12 has not looked at the last 30 years of research at the level I have, and never will. And so that's really one of the big flaws, is having a jury of my peers figuring out if something causes cancer. A jury of my peers can't agree on where to go for dinner. So it's something that I'm very concerned about going forward, that we're letting courts decide science, not scientists.
Yeah. Thanks.
Good point. All righty.
Yeah.
Well, that was an excellent talk, Kevin.
Yeah, thanks, Dave. I really appreciate the invitation. This was really, really nice, and I was really happy to be able to do it. So anytime you need any thoughts on any topic, just give me a holler.
Well, yeah, maybe next year we'll have you back to talk about GMO, and because you gave such a great talk at SciCon on that. So
we could talk about GMOs. We could talk about so many different topics.
The talc thing. Yeah, I guess that's another thing that's been blown up in baby powder.
Yeah, that's another one. Johnson and Johnson is a massive company with deep pockets. Going after them, and how easy would it be for any of us to convince 12 lay people that someone suffering from a terminal illness is should get some compensation from a company where maybe they did something wrong. You know, a massive company, and the deck is stacked against the corporations. And I'm no big fan of big corporations. I'm a scientist who works for the public, but at the same time, we really have to stand up for the science over these kinds of things. So, any help any of you can do on this is fantastic. So, thank you for your time.
All righty. Well, thanks for coming.
Yeah, thank you.
I had one more thing to show, which was that sunrise video that I tried to show last month and just couldn't because of Windows computer and Zoom, and
Yeah,
it
let
was
me
just all too much. So yeah, if you could release the screen sharing. There we go. I could find that frame lapse. Here we go. Oh, I hope you're seeing it. Okay. So this is 60 times normal speed sunrise here in Socorro, and you can see the shadow of the Earth.
I think you have to share your screen. Yeah, we're not seeing it, David.
You're not seeing it.
No, you got to share your screen.
Oh, darn. Okay. So let me try it again. It was working great for me. Okay. I'm going to share desktop. See a movie now?
That looks good.
Yeah. So it's only at the end of this video does the church and the trees in the local area finally get lit up, which is exactly backwards from the flat Earth. If things that are closer to the sun get lit up first, then it should be in the reverse order as shown. So hey, that worked great. So we got a good system for moving forward. Thanks ever so, Kevin. That was great. And we're still working on next month's program, but we'll come up with something. One more note on having meetings in person. I did check on the Copper Canyon. Not only did they close at 3 p.m., but they're closed all day Wednesday. So that is a non-starter. But we used to meet at UNM, and then we left because they started to charge too much money. But we have a budget, and we could easily afford $7,500 to have a meeting. So I think maybe we should look into that, because the restaurant is a pretty close environment for getting people, without masks, sharing all kinds of respiratory fluids. So, a classroom where people can spread out might be preferable. So I'm going to look into that. And maybe we'll revise our online poll to take a second go at it. So that's all I had. Anybody else have anything before we turn you all loose? Okay, well, have a great evening, everybody, and have a great month, and we'll see you next time. All right, thanks, Kevin.